because you and me, the normal eyeball, I think no problem. If someone have the defective problem in the eye, so it means there is a developmental problem for the child. That's what they have some problem in the eye. Okay. So I think the secretary mentioned in the Bhopal, the gas, some of the eyes are being protruding out, right? So like that, I think that is a chemical effect. That is different story. But even the defective, gene defective, there may be a chance to have some problem. So what I'm trying to say is, some of the genes is responsible for the eye development. For example, PAX6, PAX6, write down, PAX6. And then you just even go and refer that particular PAX6 gene is responsible for the development of the eye. Okay, suppose if there is any mutation, it means what it will do during the development, the cells, some of the cells it is neurocrystal origin or some other epithelial origin like that. So some of the cells, it is considered a neurocrystal origin. So those cells, it will come and then it will come to the globe. It will come to the globe here, and then it will develop the proper structure, either the cornea or the iris or the lens. It will form the proper development of the particular organ or the structure of the eye. Okay, if there is any mutation in the PAX6 gene, okay, so the migration of the cells into the globe is a problem. It will never never migrate to the globe, and then there is a problem in the structure of the eye. So that is not giving the right visual ability. Okay. Okay. So I think I don't go in detail. So these are the disease, and these are the candidate genes for each and every genes. Please, I will emphasize once again. If it is a disease, one disease having only one gene. For some disease, it is having many genes. Okay, one particular gene is mutated, it will cause only one particular disease. For some other disease, many candidate genes are there. Many candidate genes. See here, the cataract, so many genes are there. It's a very complex. Very complex. That's what we are screening, either 600 genes or 19,000 genes. Okay, so all the thing, so we studied entirely either the single gene or multi genes, and then we come up with what is the exact mutation happening in the particular disease or the particular patient. Okay, so and then this is the diabetic retinopathy. The diabetic retinopathy is very common now. It means we are not maintaining our sugar level, and then many people are being diabetic. But one group of people develop the retinopathy, one people are not able to develop retinopathy. So why? They are diabetic, but one is developing retinopathy, one is not. Again, that is depending upon the genetic makeup of the particular person. Okay, even he exposed to diabetes for a long time, he never developed retinopathy. But some people know immediately. Maybe you can go and refer your mom and everything. Some of the diabetes patients immediately after 5 years, after like 50 to 55 or 50 to 60, they will lose the vision within 5 years. But some people, they will live longer, 80, 85, no problem. So it means there is a specific genetic marker is there to develop the retinopathy. That is the point. Okay, see here, this is a blood vessel. It's a very fragile one. In the advanced stage, even if you sneeze, I'm talking about the high end. If you sneeze, inside the eyeball, there is the blood vessels are very fragile, and then it will burst, leak the blood, and then it will completely, within a short period of time, you can't able to see. Okay, so this is a very complication. What I'm trying to say is, the patients, if it is a diabetic patient, they will go to only the diabetic clinic. They never come to the eye hospital. They may prone to develop the diabetes. So you have to take care of your eyes too. Okay, so this is we identified what is the specific genetic marker for Indian population or Southern Indian population. So this is one of my PhD students. Right now she is in California. Okay, so she is the entire PhD thesis has been published in BMC Medical Journals. And then we identified, yes, there is a candidate gene for a specific genetic marker for diabetes. Okay, so now I think people are donating the eye, right? After donation, after the death, within the 3 hours, you nucleate the eye. Just pull the eyeball, cut the optic nerve, then you have the eye globe, and then put it in a specific solution, and then we will take only the cornea. The cornea, this is the cornea. The cornea you can transplant to the other patient. Suppose acid burn, alkali burn, Stevens-Johnson syndrome cases. These are the cases. So you can transfer whatever the cornea you collected from the eye, from the gonated eyes, and then you can transform to the patients and then get benefit. Okay, some of the movies have been taken based on this too. Some movies. Okay, so based on this, now the epithelium, stroma, and endothelium. Okay, so this is the layer. So epithelium, stroma, and endothelium. These are the three main layers. Okay, so the epithelium is a multi-layer, and then the endothelium is a single layer. So here, the epithelium, the epithelium. Suppose consider there is a wound in your hand or leg. It will heal immediately. Okay, so if there is any problem in the nerve cell, okay, it will take long time to proliferate. So the same scenario here. If it is the endothelium is gone, that's it. They have some problem, and then there is a visual acuity. They will be able to see problem. Understand? So again, there is a gene behind it. If it is a gene is perfect, it will form the multilayer. If it is a single, the epithelium genes, if it is a defective, it won't form the endothelium. Okay, so till 2006, nobody knows the candidate gene for endothelium. Nobody knows. Okay, fortunately, in Aravind, we collected 5 families, and there are 2 families from Singapore, and there are 2 families from Myanmar. So all the samples we did together, and then we did the linkage analysis. Linkage analysis in the sense, you take father, mother, grandson, grandma, grandpa, all the samples collect, and then you do a genetic analysis, linkage analysis, which marker is transferring from grandfather to the 5th generation. So if we entirely sequence, and then you can able to identify which gene, where is the mutation, where is the change, and how it is inheriting. Based on that, we identified first time this is the candidate gene for this particular disease. The disease name is, so this is the condition. See, it's a very, very rare. It's a very rare, but you can say it is a rare to the Western population. But in our India, our population is huge population. 3,000 patients, we are seeing in Madurai itself per day. I think even if it is a rare disease, we can be able to collect lot of samples for this kind of thing. You may think, see, your business is good, you don't need to worry about others. You think, according to this particular child or the parents, how they have painful, within the family. Okay, so you have to work on that. So here, I think, see here, this is the epithelium and this is the endothelium. See, the endothelium layer is completely gone this for this child. Okay, and then, as I mentioned, we identified the candidate gene for this particular disease that is present in the 20th chromosome. We have 23 pairs of chromosome, 22 autosomal, and then one XY chromosome. Okay, the female XX and the male XY. So here, this is the 20th chromosome. This is the name of the gene, and please recall the structure of the gene. Okay, so this is the exon intron like that. So these are the this is the variant should be there, but consider this arginine, R for arginine. So it is converted to truncated. X means that is the complete protein synthesis stopped in the particular region. That's what they have the disease. Okay, we identified exactly what's the variation in the gene, and then you have to functionally prove. You have to functionally prove not only the screening, and then what you have to do, you take that particular plasmid. Okay, plasmid, you clone the normal SLC411 gene, and then you compare where the mutation happened, and then what is the difference between the wild type and the mutated type. See here, the wild type, the protein is perfect, and then here the mutated one is behaving like this. So based on this, yes, the protein is truncated in the particular region. Okay, and then here this is the immunohistochemistry. So the protein, the genes inside the protein, that is the true gene. So some of the proteins is expressed, it is extracellular or intracellular. If it is intracellular in the sense, the protein, even if it is truncated, it will sit inside the cytoplasm. Some of the proteins come out of the cytoplasm. That is depending upon the gene. So here, we know where it is coming, whether it is sitting inside or outside. So based on that, using immunohistochemistry, how it is behaved between the patients and then the normal. So based on that, we proved this particular gene is responsible for the disease. So this paper is published in Nature Genetics. Okay, and then this is one particular disease. One is given a very common disease that is called, I think they don't like it, Have a nice day, guys. Okay. This is a